All right, today's podcast is talking about Mendelian genetics, and then we're going to explore it further. We want to explain the principle of independent assortment, describe the inheritance patterns that exist aside from simple dominance. These were more complex traits than what he saw in his original experiments, and explain how Mendel's principles apply to all. Now, we're not sure exactly why he didn't look at these traits first, but it makes sense when you consider that they're more complex. And so he took the simpler, it seemed, the simpler traits first. So Mendel's next question was, does the gene that determines one trait affect the gene that determines another trait? Like, for example, uh, remember that we talked about round and the round pea versus the wrinkled pea, and the yellow pea versus the green pea color. Okay, well, he wanted to test this. So to test this, he crossed the purebred round yellow seed plants with the purebred wrinkled green seed plants, kind of like this. So we see the round yellow, big R, big Y, big Y. Notice that when it makes gametes, you're going to have one of each of the traits. Then he took the wrinkled green, the recessive traits, and once again you get one of each. So he, when he did this, he the expected 100% of the round yellow seeds came up. Okay, but the question is, would a cross of the F1 generation, the offspring here, would they have a similar ratios to a single trait cross? Remember, a single trait cross, it ended up being 3-to-1 of the dominant trait in the phenotype to one of the recessive traits. Okay, so would it be similar to that? So this shows his uh, what he got from the F1 cross when he took the the offspring of the first generation, the parental generation. And in the plants that were produced, there was a ratio of approximately 9 of the yellow round seeds to 3 of the green round seeds. So 9, 2, 3, 4, 5, 6, 7, 8, 9, and then 3 of the round green and 3 of the yellow wrinkles. So these have a dominant of the yellow, but the wrinkled recessive. These have the smooth dominant, but the green recessive. And then the wrinkled, there was only one of them with the green wrinkled, which was both a doubly recessive. So the result demonstrated that at least for the traits that Mendel chose for his cross, the genes assorted or segregated independently. So your question is, for number 17, what is independent assortment? And independent assortment, as it says, is the genes for different traits can segregate independently during the formation of genes. So the for traits can segregate independently during the formation of gametes. So in other words, these two alleles are going to separate from each other. Now this isn't always the case, as we'll show you later on. So here's a summary of Mendel's 4 principles. First of all, genes are passed from parents to offspring. Some alleles, different forms of a gene, are dominant and some are recessive. So like the green is recessive to the green pea is recessive to the dominant yellow. And most sexually reproducing organisms have two copies of, of each gene, one from each parent. Genes segregate or separate during gamete formation. And then alleles for different genes usually segregate independently of one another. We call this the principle of independent assortment. Give you a chance to write in. Oh, by the way, I'll put this up on Moodle so that you can, if you miss anything, you can go back for it. So beyond dominant and recessive alleles. So some alleles are neither dominant nor recessive, and many traits are controlled by multiple alleles or multiple genes even. So if you look over here on the right, this one right here is an example of a cross in which here's a red snapdragon cross with a white snapdragon, and notice that the offspring all come out pink. But they're not blended the way that they used to think they were, because if you take this offspring from the original cross and you put them back together, you end up getting some that are red, some that are pink, and some that are white. So it's not a true blending. Uh, we call this incomplete dominance, when neither allele is dominant. So the red making gene is, or allele, is making some red, while the white is making some white, and they don't over, neither one is dominant over the other. So the heterozygous offspring are intermediate between the two homozygous phenotypes. So heterozygous, meaning that they have one of each of the alleles, are intermediate between, or between the two homozygous phenotypes. So, okay, 20. So beyond dominance, we had, so we talked about incomplete dominance. This is called codominance. Now, when you have a co-captain, you have, they share power, okay? So neither one of them are dominant to each other in this case, the two alleles. So codominance, both alleles for one trait contribute to the phenotype. The heterozygous offspring expresses both traits. So here is a red horse that crosses with a white horse, and you end up with what we call a roan horse. So you don't just have um, red, or you don't have pink, you have red and white hairs that are equally distributed throughout the body. Okay, so that is called codominance. So now the question is, what is the difference between incomplete dominance and codominance? Remember, incomplete dominance, the two traits that were expressed were half, or they were intermediate between each other. So the red and the white flower made a pink flower. Okay, the red didn't overwhelm the white, so it showed up pink. Or in this case, for the codominance, you have the red and white hairs. You don't have pink hairs, you have red and white hairs. Okay, so it's Just not a very big difference, but it's an important difference because in the incomplete dominants, they both make something that doesn't overwhelm the other. In the codominants, both of them actually are made. Each of both of the traits are made, and neither and both of them show up. Okay, this is going to be important later on when we talk about blood typing. That's another example of